Algae is an aquatic plant that falls under the kingdom Protista. They contain photosynthetic pigments that allow them to self-feed and assume the role of one of the main primary sources of energy on Earth. They store this energy in the form of carbohydrates. Algae is responsible for making up to 330 billion tons of oxygen each year, which is approximately 70 to 80% of the oxygen in the atmosphere. Algae typically absorbs light at around 430 nanometers and 662 nanometers, which are blue and red light respectively. The amount of light algae absorbs is dependent on the concentration of chlorophyll A and various other photosynthetic materials. Nitrates are important ions that allow plants to grow and produce flowers. Typically, plants absorb nitrates in water through their roots, and some obtain this nutrient through human-made fertilizers. The following graph shows chlorophyll A density collected by 3 separate lab times in 3 areas. 4 rocks were used per location, except for the San Jacinto area because of miscommunication. The overall average algae density, as recorded using the chlorophyll method, is equal to 0.0249 milligrams per centimeter squared. And you can see the rest of the results on the following graph here. One of the biggest issues with this data set is that most of the math was incorrect and not converted to the right units in the beginning. In conclusion, there are too many errors, and therefore the data is unreliable. The following is the level of nitrates measured by the various researchers in various locations at various times. Here, this graph demonstrates the correlation between nitrates and algae growth in 3 locations. One of the main issues with collecting and plotting this data was that between the 2 experiments, only 3 locations related to both. Therefore, because there are so few points, a reliable R-square value could not be calculated. Chlorophyll and ADFM measure algae density on a different scale. They cannot be used on the same side of the axis. However, since they tell the researcher the same information, one may be switched out for the other. In this chart, chlorophyll data is used instead of AFDM in comparison to the level of nitrate in the water at Waller Creek. The results found above were all founded within the same study, but relied on the collaboration of more than 50 FRI students. For the Sunny chlorophyll experiments, the 3 groups obtained standard deviations of 0.0443, 0.48633, 0.3916, respectively. Group 2 showed the highest amount of standard deviation, but regardless, all 3 groups demonstrate low variance. And finally, the standard deviation for the shady chlorophyll group was also low in variance at 0.039, 0.0006, and 0.1833. The low variance in each individual group data shows that the data collected hovers around the average and is more or less accurate. For the nitrate experiment, the same group of FRI researchers collected their data at various locations and times. Nitrates were collected in 3 different trials. However, since the researchers did not consistently fill in the data sheet, only the first trial, the most filled section, was used to keep the numbers consistent and protect against unforeseen outside variables. By comparing the average algae concentration to the nitrate levels in the same location in Waller Creek and formulating an R-square, the researcher is able to determine whether or not the 2 factors have any relations to one another. The R-square value obtained from this data set is equal to 0.022. A R-square value this small shows little to no correlation between the 2 sets of data. It can therefore be concluded that the level of nitrates does not affect the level of algae concentration, which means the null hypothesis was proven. However, as mentioned before, because of so many mathematical errors and inaccurate data, as well as unaccounted variables, the results are not reliable. If the experiment was ran once again with accurate and consistent methods of collecting and recording data, future experiments based on the previous experiment would be to find the source of nitrates using the concentration of the algae. Or if the null hypothesis remained true, then it may be more accurate and the researcher would have a higher level of confidence. Afterwards, a cuvette is filled with 90% ethanol and another is filled with algae extract. A spectrometer is then used to measure the optical density of the algae extract at a wavelength of 750 nanometers and 664 nanometers. Next, the algae is acidified and the optical density is taken at those wavelengths again. Finally, to find the chlorophyll A concentration, the following equation is used. These following materials were used. The Environmental Integrity Index experiment, which the nitrate concentrations were recorded, used the help of more than 50 researchers performing various tasks and inputting their data collectively into one spreadsheet. To collect the amount of nitrate in the water, a V2000 was used. In order to use the V2000, the number I22 program key must be pressed. Next, the researcher inserts a blank vial into the V2000 in order to zero the reading. After blanking, 25 milliliters of creek water is mixed with the green capped reaction tube. After adding 10 drops of the A69OI acidifier solution into an empty cup in the zinc foil pack, the solution is put into the reaction tube and shaken for 2 minutes. Afterwards, the nitrate vacuum valve is inserted into the sample cup tip first and snapped open. When all the contents are mixed, place the vial into the V200 to obtain the nitrate reading for that selection of Water Creek. Finally, in order to obtain the final results, Google Spreadsheets was used to combine and compare the data of the two experiments.